Well, thank you very much for that gracious introduction. I'd like to talk about several things today. One, I'd like you to understand that systems medicine and P4 medicine, which I'll explain, are fundamentally different from how we practice medicine today. And two, I'd like you to get some sense of how wellness is really going to transform healthcare in the future. And in fact, wellness is going to create an emerging new industry with enormous innovative opportunities for company creation. So let me say that over the past 4 or 5 years, I've been involved in starting 3 companies that all play different kinds of central roles in systems medicine: a blood diagnostics company, a company that is actually going to create reagents that will replace monoclonal antibodies, and a wellness company. And I'll talk about all 3 of those in the context of this lecture. When I started at Caltech in 1970, the question that really plagued me was this one of complexity. Biology and medicine, disease, both were incredibly complex. And the tools we had at that time, molecular biology, cell biology, thinking about genes, one gene, one protein at a time, just weren't going to cut the complexity. And I think the analogy that's really a good one is the 6 blind men and the elephant, each feeling a different part and making a declaration that the elephant is a spear or a fan or a tree. And the elephant really is these and much more. And so it is with complex systems. And in fact, the initial problem we had is we didn't have a conceptual framework for thinking about complexity, and that later came to be called systems biology, and we'll talk a little bit about that. But what I was also struck by is when you thought about complex systems, we had neither the technologies, again, this was 70, nor the strategies for dealing with all of this complexity. And I remember reading a fascinating book in 1973, Thomas Kuhn's The Structure of Scientific Revolution, which talked about paradigm changes in physics. And he made 2 major points. One, that paradigm changes are really hard because you alter the way people think, and very few people can think out of the box to create the initiation of a paradigm change. But he said even more fundamental is the enormous conservative nature of scientists and their reluctance to give up long-held beliefs. And it was with that in mind that I had the good fortune over 45, 50 years of my career to participate in a series of paradigm changes that I think really did transform biology in many ways. So one was bringing engineering to biology. We developed 6 instruments that let you read and write DNA in various ways. And what these instruments did was, one, opened the era of high-throughput data gathering in biology, and 2, they ended up ushering in big data and its analytics, and you'll see how critical both of those things are. One of the instruments, the automated sequencer, got me involved in the second paradigm change. I went to the first meeting held on the Human Genome Project in the spring of 1985 with 11 others and debated the pros and cons of the Human Genome Project, and we came to 2 conclusions. One was it was feasible, though technically really difficult. And the second was the 6 of us were really split 6-6 on it, and the 6 who were against it were really fanatically against it. And the essence of it was big science is bad science, and it will destroy small science. And when we went out in the world, 85, 86, 87, I would say initially 80 to 90% of the biologists were opposed. And the institution that was most opposed to the Human Genome Project for the next 5 years was the National Institutes of Health. And it reluctantly gave in when a National Academy Committee powered by half opponents, half proponents unanimously endorsed the project. And of course, what the Genome Project gave us from the point of view of systems thinking was a complete parts list of genes and by inference proteins. So for the first time, we could start thinking about systems approaches to human biology, and of course the biology of other organisms whose genomes were done. The other thing developing the automated sequencer made me realize where we had to put together engineering, chemistry, computer science, and molecular biology was in the future, the hard problems of biology were absolutely going to mandate living in a cross-disciplinary environment where you brought together all sorts of different flavors of scientists. So biology could effectively drive relevant technologies, and the technologies in turn drive the analytic tools. And if you could put all those things together, you really could transform biology. So I proposed to Caltech in 1988-89 to the Caltech president that I'd like to start such a department at Caltech. And he was really enthusiastic, as were the engineers and the chemists, and physicists were indifferent. But the biologists were absolutely opposed. So Bill Gates made it possible to go to the University of Washington and start such a department. And what was really spectacular is in 8 years, that department created the first two critical technologies for the then emerging field of proteomics. It created the first two analytic tools for the Human Genome Project, assembly, quality control. We developed the inkjet technology there for large-scale DNA synthesis that Agilent has commercialized, and on, whole series of successes. But what was interesting was the dean who hired me said at the end of 4 years, he'd give me another floor of a building and I could build on top of the cross-disciplinary department, systems biology. And as serendipity would have it, that dean died just at 4 years in an avalanche in the Himalayas, and the next dean had a very different point of view. And I decided, as a consequence, in 2000 to resign, start the Institute for Systems Biology, and of course that opened up system science and how we could apply it both to biology and medicine. And that in turn led to systems medicine, P4 medicine, all those things that we'll actually talk about in just a few minutes. But I will say that the two lessons I learned from going through these paradigm changes are, one, scientists are indeed incredibly conservative. Every one of these proposals that you have before you met enormous objections. I remember when I started the Institute for Systems Biology, a really famous friend at MIT said, systems biology is hype and trivial. It'll never amount to anything. And he led a National Academy report 10 years later that said, systems biology is the future of biology. So it's great to see even conservative scientists can actually change. But I think, determined optimism is really critical. If you really believe in something, don't worry about what other people say, just do it. The more important thing I learned, though, is for each really innovative idea that one has, paradigm change, it is extremely difficult to have them emerge in the context of a given institution. Bureaucracies are formed by the past. They just barely let you deal with the present, and they mostly can't handle the future. And so each one of the first four of those things, we required independent new entities to actually execute them. And I think one of the fascinating questions is what's going to happen with systems medicine, P4 medicine. That, of course, is an unanswered question at this point in time. So I would say systems medicine does really employ system strategies. And I think there are two really central points about systems medicine. One is the idea that an enormous amount of complexity has to be dealt with by big data. So what we're going to see, and we've already seen it, I'll tell you about a project that's done this, is that each patient is going to have a virtual cloud of billions of data points, many different types of data, and we'll have the computational tools to integrate and model the data to create and identify actionable possibilities that can either improve wellness and or let you avoid disease. And of course, the really critical question in big data is signal-to-noise issues. And to solve the problems of big data, it isn't enough just to have clever machine learning. You have to integrate with that deep domain expertise. And I'm sure we'll hear about that from the next speaker. One way to reduce enormously this data dimensionality is to convert it into biological networks. These are the information conduits that regulate development, physiologic responses, aging. And of course, when networks become disease perturbed, they mediate disease. If we can capture the nature of these disease perturbed differences from their normal counterparts, we can understand in a deep sense the pathophysiology of the disease and get new insights into diagnosis and into therapy. So by about 2006, we really had very clearly articulated the vision of systems biology and started to articulate the vision of P4 medicine. But one of the things that I was struggling with is how could we really bring it to the healthcare system so it really made a difference? And I will say in those early years, we had a number of efforts that all turned out to be absolutely ineffectual. And in retrospect, it was really clear why each of them was ineffectual. And then in 2017, what happened is I met the Minister of Finance for the state of Luxembourg. He was interested in changing the economy's dependence at the 90% level on financial services. And he wanted to bring in healthcare and biotech, so he asked us to write a proposal. We did for them many things, which I won't describe, but what they did for us was to give us $100 million over 5 years. And what this let us do was two really important things. One, we could take on high-risk, high-gain projects, the technologies, the strategies of P4 medicine, systems medicine. And number two, we had the money, which was all basically flexible, to actually finish these projects in a way that could bring them to the healthcare system. So we developed 9 or 10 strategies and technologies. I'll show you some of those in just a moment. And it placed us in 2013 or so at this incredible tipping point because it was clear we could really transform how we thought about many critical issues, including diagnostics in medicine. What we did then was proposed in 2013 to take on the longitudinal analysis, creating those dynamic data clouds I spoke about, of 100,000 well people to begin to understand what it was to focus on individuals in the context of medicine. And what we've done over the last year for a 10-month period is taken 107 individuals as a proof of principle, and the results are spectacular. We'll talk about those at the very end. And of course, this fits in with Obama's initiative, his proposal on personal medicine or precision medicine. If it's defined properly, the definitions right now are very vague at NIH. I will say, I went to Francis Collins early when I was setting up this 100 Pioneer project, and he said, under no way would NIH ever support a project like this because it was so far outside the bounds of what it did, and he needed his money for good science that was sitting there waiting to be supported. So I guess he hadn't heard about precision medicine yet. Or maybe he has a very different definition. We don't really know. What we did was develop a lot of technologies, and I'll only talk about one of these. There is an incredible company that has come to ISB from Intel that basically learned how to take two plastics and create pores of very controlled size. And what they've done is they figured out how to put a nuclease on the lip of such a pore, capturing a strand of DNA, cleaving off the nucleotides, sending them through the pore, and then electronically detecting the unique signatures each of the 4 bases produce. So they've demonstrated all of those things. Now, what is interesting about this technology is, number one, nucleases are infinitely processive. So you can really think about 100,000 kb reads. And that will transform the assembly of human genomes, which is one of the things that plagues us today and doesn't let us get indels very effectively. Number two, it's clear that nucleases operate equally effectively on DNA or RNA, so you can sequence RNA with this every bit as well as DNA. Number three, because each of the 4 bases gives quite unique electronic signatures, one can speculate that the 16 covalent modifications, the epigenetics of covalent modification of DNA itself, could be read off at the same time you do the DNA sequencing. And finally, number 4, it is clear that one could parallelize this process to the point where you could think about doing a 15-minute genome. And my prediction is, if this company gets the funding, there are technical limitations I haven't talked about, but I think in principle it could be done. I could see a human genome being $100 in a period of 5 years or so. And we did a lot of other technologies, and I'll talk a little bit later about one of those. But we developed a series of strategies, too. One was a strategy where we could look from the beginning to the end at the dynamics of disease, in this case using animal models, and I'll show you an example. And the second is we developed systems approaches to blood diagnostics that have given us enormous confidence that for any clear phenotype difference between normal and diseased, we can create blood markers. And I think proteins are going to be by far the most effective blood markers. And then finally, we'll talk about longitudinal studies. We did a lot of other things, but we really haven't got time to talk about them. In mice, we induced neurodegeneration by injecting into the brains of mice activated prion particles in C57 black mice. This was a 22-week, it was a disease of 22 weeks progression. What we did was compare the transcriptomes of the diseased animals with their normal counterparts at 10 times across that window, so we could look at the dynamics of how the brain transcriptomes changed. And what we were horrified to see initially was 7,400 genes changed. That's a third of the mouse genome. But most of that was biological noise, that is, it was created by irrelevant biology. So we created 6 inbred prion strains of animals that allowed us to subtract most of that irrelevant biology away, and we were left with 300. We were reasonably sure represented the core of what the neurodegeneration process was about. We did serial histopathology on these animals and demonstrated 4 major processes that were operating: prion replication, accumulation, and glial activation in 2 forms of neurodegeneration. Each had partial protein-protein interaction networks. We mapped the genes and their dynamics into those networks and came to 2 really striking conclusions. The first was that the 4 major networks were sequentially disease-perturbed, starting with the most unique. And the reason this is key is it's that first network that'll really be the key to reversing the disease once you understand the pathophysiology, the networks that mediate it, and can manipulate them with drugs and, of course, the appropriate early diagnostics. The second thing that was fascinating is only 200 genes mapped into the 4 major networks. The other 100 identified 6 smaller networks. No one had any idea were a part of this neurodegenerative process. But if you looked at the dynamics of each of the 10 networks, They explain virtually every aspect of the pathophysiology of the disease. So that's why following the disease from beginning to end, I think is really going to be key. And one of the questions we'll discuss is, how can we do this in humans? And there is absolutely a straightforward way we'll talk about to be able to do it. We've done this for a whole series of different diseases in animal models, and I'm, again, not going to discuss them, but I will say in each, the principles of the dynamics are very similar. In each, there is a network or a series of smaller networks that initiate the whole process and give you insights into what you should be thinking about if you want to terminate the disease process early. So we think we've really made blood a window into looking at distinguishing health from disease. And in 2012, there was a beautiful report written by the Institute of Medicine that surveyed all the preceding literature for 5 or 6 years. And they said, isn't it interesting? There are about 400 plus examples of presumed diagnostic markers that were papers in Cell, Science, Nature, etc. Not one of them made it into the clinic. So the question they asked is, why is that? And there were 2 really great reasons. So one is, if you compare normal bloods and disease bloods, you see there are enormous numbers of differences, and 99% of that is noise. How do you separate very small signals from the noise? Most people don't have any idea how to do that, and you really do need systems approaches in one sense or another to do that. The second point, which was really telling, is that there's enormous genetic polymorphism in the humans. So if you optimize the diagnostic in one population, like as not when you go to a second population, you'll go from 95% sensitivity and specificity to 50. So what it means is your studies have to begin with people from multiple geographical areas, so you subtract out all the polymorphisms. And we did one study on lung cancer. I'm just going to describe. I'm not going to go into the details, but we followed all of those principles. So what's key about diagnostics? I think blood is the ideal venue. All organs dump proteins into the blood. I think doing longitudinal studies, I think having multi-parameters so you can sense multiple potential disease-perturbed networks. I think being able to do things quantitatively, I think proteins really are going to be important. And I'll tell you very briefly about 2 approaches to diagnostics. One is using organ-specific blood proteins, and the second is using various filters to get rid of all the noise that's present in the blood. And we'll give an example of each. So in the first case, we actually used brain-specific blood proteins to look at the Prion disease animals that I talked about earlier. So the idea of organ-specific proteins is to do a deep transcriptome comparative analysis of 50 organs in humans and 50 in humans and 50 in mice, and then to identify transcripts that are relatively uniquely present in just one organ. Then you have to look at the proteins those transcripts make and see if they can be detected by targeted mass spectrometry in the blood. And we did that with humans, and we have more than 200 brain-specific blood proteins that we can see. They map into many of the major networks in the brain. And so that means a normal brain has all of these 200 proteins that at one level, if it becomes disease-perturbed, those proteins whose cognate networks become disease-perturbed change their concentration levels and do so in a way that's specific for each disease, because each disease generates disease perturbation in different combinations of networks, so you can distinguish health from the disease. So we took 15 mouse brain-specific blood proteins that map pretty evenly into the 4 major networks, and we're able to do in the blood 2 important things. One, we could see changes in the blood at about the same time we saw changes in the brain transcriptome. And you can ask, how is that possible? And it's possible because networks really record interesting amplifications of modification and change. So we could do early diagnostics. Number two, we actually in the blood could follow the successive disease perturbation of each of the four networks so you could follow progression. And we'll talk in just a minute about what that means in a more general sense. Now, the other thing we did with the company integrated diagnostics was to use these filtering strategies. Anyone who's interested, I'll explain them in detail, but we don't have time to talk about it right now. But the fundamental question we were interested in is in the States. There are 3 million nodules in the lung a year, and how does a pulmonary oncologist decide what to do? And of those 3 million, 600,000 went to various surgical procedures. And of those 600,000, more than half the procedures were done on people with benign nodules. Unnecessary cost, unnecessary morbidity. So what we set out to do was to develop a diagnostic that could distinguish benign from neoplastic nodules in the lung. And we identified 13 proteins in the blood that actually had the ability, and now we've moved it up to identify unequivocally about 50% of the nodules. And the reason that was important from the payer point of view is that saves the healthcare system $3.5 billion a year. So this is the first protein panel, we were using a CLIA lab to generate the results, ever put forward, and it was commercialized in an incredibly short period of time. And we now have 5 or so payers that cover roughly 500 million lives, and these people bought into this because of the savings as much as because of the benefit to the patient. Question you can ask is, what are these 13? We started with 400 proteins. We ended up with 13. So what do they do? 12 of the 13 map into disease perturbed networks in small cell lung cancer. So we'll have the ability both to do early diagnostics and follow progression. We're just working on those kind of things now. So these kinds of approaches let us distinguish normal from disease. They let us do early diagnostics. They let us follow progression. We'll be able to follow response to therapy. In cancer, you'll be able to see reoccurrence. But really importantly, you can identify the networks that are disease perturbed, and they themselves provide interesting therapeutic and diagnostic targets and so forth. And we're quite confident we can use these to stratify all types of lung cancer into their distinct groups. And stratification is one of the most important aspects of diagnostics and saving money in the future. So finally, let's talk about reagents that are going to replace monoclonal antibodies. What a bold statement. This has been pioneered by Jim Heath at Caltech. We started to do this in integrated diagnostics, and then more recently we spun off a company called Indie Molecular that is creating these reagents. And the idea is the following. You create a library of circular 5-mer deamino acid peptides of 10 to the 7th or so. And then you can take the protein against which you'd like to make the capture agents, and you can find monomers that bind at low levels. And you can use the type of chemistry called click chemistry to join 2 monomers together in their proper 3-dimensional orientation. To make a dimer, we've made trimers and we've made tetramers. And with each of these mers, there is close to a log increase in sensitivity. So for circular peptides, the dimers we select for the most part have the same affinity as some of the best monoclonal antibodies. These reagents are incredibly stable. Jim put the first one we made in his trunk of his car in Pasadena in June, and in September we took it out and the affinity was unchanged. So that means you could put these reagents in an envelope, you could send them to Africa, and you'd have perfectly good reagents. And of course, the other thing that's really interesting is this is all chemistry. And it can all be done in vitro. If you know the two monomers plus the nature of the coupling structure, you can use copper catalysis to assemble as much of the reagents as you want. So it changes the ballgame in many different ways. Jim has demonstrated more recently that he can focus the action, the interactions of these peptides, these protein capture agents on single peptides, and in doing so, he eliminates most of the cross-reactivities that plague antibodies, particularly when you're trying to analyze complex mixtures like blood. So we've also demonstrated these are very small molecules, obviously, 4,000 Daltons, that kind of thing. They're absolutely beautiful in vivo reagents, and here's an example of an anti-PSA that had a copper on it that we could look at with PET scanning and show localization in the mouse to the prostate absolutely beautifully. And what that means is we can look at spatial dynamics as well as temporal dynamics, and both of those are important in dealing with signal to noise in systems medicine. And without going into any detail at all, Jim has recently shown these are beautiful reagents to identify and block the activity of molecules that have been hard to drug. They work beautifully in blocking protein-protein interactions, and they very nicely modify allosteric changes. So they're going to be absolutely terrific kind of drugs in the future. So they're stable, they're sensitive, they're digital. We can make them in vitro. We've eliminated a lot of the cross-reactivity, and we feel we can expand this out on a large scale and do in vivo, in vitro diagnosis and therapeutics and so forth. And I think these are really going to be competition for monoclonal antibodies in the next 10 to 15 years. And again, integrated molecular, Indy Molecular is really pushing ahead in these regards. So, 2012, 2013, back to the P4 medicine, now that we've talked about systems medicine, there were some really interesting convergences that came together to define P4 medicine in an interesting way. First was systems medicine. We've talked about that. The second was big data and its analytics, and in parallel, the digital revolution coming on self-measurements and things like that. And third, either social consumer-activated patient networks or patient-activated networks. And let me show you how these are important. And this slide is really important because it shows you all the ways that P4 medicine differs from contemporary medicine. It is not merely an incremental change giving you drugs that can deal for a very short period of time with cancer. I have to tell you, I'm really skeptical about sequencing cancer mutations and getting drugs for them. Let me just tell you, do you know there's no epithelial cancer that's ever been cured by drugs? I think the key to curing cancer in the future is going to be immunotherapy, so you really ought to start thinking about that. The best drugs can do now is let the patient live for an extra 6 or 8 months, but they become resistant, and you're back where you started again, but you've spent $140,000 or something like that. I'm not sure that's a wise use of society's resources. So P4 medicine is proactive, not reactive. It is all about focusing on individuals and not on populations. It's a major focus on wellness, which has been virtually ignored by the current healthcare system. It's all about creating these dynamic data clouds that I talked about before, and it takes a very skeptical view of therapeutic trials where you put together 30,000 people and you give them drug or placebo and you extract information. Every patient is unique genetically, unique environmentally. You have no business averaging that data. And you do a marvelous job of suppressing signal and increasing noise. And drug companies have shown that. So what you really need to do are these N of 1 experiments with individuals. Do it with 30,000 individuals, and then aggregate the individuals according to the properties that you're interested in. So N of 1 experiments are really going to be important in the future, and the statistics will come by generating appropriate large numbers. N of 1 experiments can give you deep and fundamental insights into where you need to go in medicine. I'll show you an example or two of that. Finally, the patient social, consumer-activated social networks are all about enabling consumers to learn about the new medicine, to do crowdsourcing with the new medicine, figure out new ways to use it. And finally, they're going to be the advocates that I think will really persuade a very conservative healthcare system and very conservative physicians to actually change. So I think P4 medicine is really about two things. It's about quantifying wellness. It's about demystifying disease. And I'll just say that 98% of society's resources go into disease, almost nothing into wellness. And as a consequence, wellness has not been studied, and it has a poor reputation. And many people say we shouldn't use the term wellness because it has this dubious reputation. And my argument is we'll make wellness really strong, and we'll use a word that's probably the best word for that thing. What I do believe is that we are now at the beginning of a whole new healthcare industry called the wellness industry. And I really think over the next 10 to 15 years, that wellness industry will exceed in market cap the healthcare industry for the disease, the current industry that we have today. And I think more and more focus is going to be put on wellness. And I'll give you justifications for that in just a couple of moments. So here's really a remarkable statistic. If the increase in lifespan continues as it has for the last 10 years, you can make a calculation that 50% of the kids born today, this year, will live to be 100. And the key question is, what are the last 30 years of their life going to be like mentally and physically? And if you look at what happens to us today, it's pretty grim for most people. So I think we really do need wellness to deal with that opportunity that's coming. And that's where I think this proposal we've made to look at wellness on a large scale is really going to be transformational. And what I'll talk about are the 107 individuals that we've put into a pilot project doing their genome. Then every 3 months, we get blood, urine, and stool to look at clinical chemistry, 700 metabolites, 400 carefully selected proteins, and every 3 months we look at the gut microbiome, and continuously we look at quantized self-measurements and get the histories and keep track of their environments and all these things. So the basic idea is to integrate these data together, and as I said, what that ends up giving you are actionable opportunities that one can actually use to begin thinking about how to increase wellness and or avoid disease. And a critical part of this endeavor is the fact that each data type can give you actionable possibilities, but even more, the integration of 2 or more data types really increases actionable possibilities. So anyway, we have people that are coaches that bring to the individuals these actionable possibilities and persuade them to change their behavior. We've had a 70% compliance, and you can look successively at your data cloud and see how your behavior has altered your data cloud in interesting ways. People divide into well and divide into transition into disease. In the well people, we can follow the extension of their wellness, and we're beginning to get metrics for wellness that will quantify wellness and what it's all about. And of course, with regard to disease transition, we can follow the data tranches back to the point of origin. We can use systems approaches to get diagnostics and therapies and convert people at the earliest stage back to wellness. So the project then, we've got a whole series of stories about what we can do. We've looked at all the transitions you see here, wellness disease to wellness, lesser wellness to greater, and so forth, and the transitions that come from the actionable possibilities. 91% of the individuals in the initial tranche had nutritional difficulties that could very easily be controlled. Almost 70% had inflammation their doctors had no clue about. In many cases, we discovered exactly what those are. We converted 7 pre-diabetics to perfectly normal individuals, and many of the other 20 or 25 of the pre-diabetics are all going in the right direction and so forth. So here's a disease to wellness transition that was really interesting. We found, looking at the genome, 2 individuals homozygous for the gene that causes hemochromatosis, high iron in the blood. One of those individuals had concierge doctors on each coast, had gotten arthritis. No one could figure out what it was. We sent the information to the doctor. He did regular bleeds. The iron levels came down, and in 10 months, the arthritis had gone. So here was a beautiful disease to wellness transition that transformed a fellow's life. Vitamin D deficiency was really interesting. Almost 90% of the pioneers were low. 6 variants in 3 genes block the uptake of vitamin D, and it causes all sorts of risks that we all know about. But if we bend the individuals according to those genes, we could show that there was a beautiful correlation between the genetics and the phenotype of the individuals. And that phenotype remained exactly the same even after we pushed the levels of vitamin D up. And we found those that had 5 or 6 of the blocking mutations needed enormous megadoses to move them at all. So, and we were able to push almost everybody into the normal level. But this raised the possibility that we could use GWAS data from many different diseases to actually begin calculating the risks on individuals. We did this for lipid levels, GWAS, 72 markers, and so forth. We mapped into all 72 individuals those markers, and we could show again a beautiful correlation in one of the families in the study between individuals with high risk for bad lipid and low levels of HDL, the good lipid, or conversely, high levels of LDL, the bad lipid. So these things, 2 of them were off scale. One was a triathlete, way off his genetic potential. The other, actually, we don't know why he had such bad results, but they were the same results as people who had bad genes. You need 2 dimensions of data to make good diagnoses. Okay, so once again, a beautiful correlation between genotype and phenotype here. And I just want to show you one more set of data that is absolutely spectacular. So we can do measurements and calculate risk in all these patients now. But here is the most spectacular data slide of all. 35,000 correlation between data types in 1 of 5 sets of data. Statistically significant positive or negative kinds of correlations. The whole dark matter of human physiology is laid out here. Many striking kinds of opportunities. So in closing, the 10-month study for the pioneers has finished. But what we're doing now is we've created a scientific wellness company. We're planning to do 10,000 people in the next 18 months. All of the pioneers have volunteered to go into that enthusiastically. Many of them feel it transformed their life. For the first time, they had control of their own health. So I urge you, as you think about what you're going to be doing here, to realize wellness is really a key point. And these data clouds, 10,000 of them will transform how pharma, biotech, diagnostic companies, and nutrition companies are going to perform their activities in the future. And finally, I would argue if you put all of these things together, we're going to see an enormous increase in the quality of medicine and an enormous decrease in its cost. And I'd argue we'll get to the place where we can even think about a democratization of healthcare where it will be brought to the poor as well as the rich. Thank you.